Hey everybody, hope you're doing well today. So I'm sorry, but I'm not going to be able to be with you on Wednesday when you go to make observations about your bacterial transformation lab. So when you come into class, things will be set up for you guys. If you look here, lab stations are pretty cleared out. You're just going to have some sanitizing wipes where you can go and clean up your hands when you're done. But what you'll do is pick up your bacteria from your particular shelf. We got period 2 on top, period 4, and then period 7 down at the bottom. So you'll grab your stack, and then you'll make observations about your bacteria. Now, again, generally the idea is it's good to keep the lid on. If you want to take the lid off for a short period of time, that's fine. Just don't put the lid down on the counter. Again, we want to minimize transmission of anything that can contain bacteria. So generally, people will have their petri dishes upside down, so lid's on bottom, and you can look at the petri dish and see if there's any growth. If you want to take the lid off again for a few seconds to look at things, you're welcome to. Just put the lid right back on. Don't put the lid down on the ground. Now, in your lab packet, if you look on page S9, it's got a couple of things. There are some laboratory questions at the bottom of S9. Number 1 says record your results and conclusions using the organizational method you devised. You know what, for this, if you just flip to page S15 in the back, it's got everything organized for you. So if you would, flip to page S15 and it will walk you through what types of things to observe. And I don't know how your stuff's going to turn out. It may have worked really well, it may not. So what I did was I just recorded an idealized version of all those results and put it up on the board. So this will be in the room for you when you come to look at things. So I'll have what you should find. Now, what could you find? Well, if you look at a plate and absolutely nothing's growing on it, we say it's a clean plate, nothing survived. So for instance, if you had a plate that has an antibiotic and the bacteria don't survive, well then it would just be a clean plate, nothing on it. Another thing that could happen is you get colonies. With colonies, they're just little dots. What it means to see a colony, it means that one bacterium took up the plasmid and survived, and now it has millions of descendants. So when you see a colony, that means one bacterial cell survived, and then it has a bunch of descendants there. Okay? So colonies means bacteria survived, but only some of them survived. Only some of them have the plasmid. If you see a lawn, you don't see individual dots. It just sort of looks like a film covering the petri dish. So that means nothing was selected against. Everything survived. So under what sort of circumstances we get situations like this? Well, plasmid 1, that has 2 genes in it. One, it has the ampicillin resistance gene, so it survives in a plate with ampicillin. 2, it has a gene for the green fluorescent protein. So if you're using plasmid 1, ideally, in your amp plate that was plus plasmid, you would see colonies. You'd see little dots. Let's see, on the minus plasmid plate, that means you didn't put the plasmid in there. Well, the ampicillin would have killed those bacteria, so it would be a clean plate. On the kanamycin plates, these cells don't have a plasmid for resisting kanamycin, so that antibiotic should have killed them. You would have had a clean plate. And here on the LB plates, with LB, it's just food. There's no antibiotic. So ideally, you would have gotten lawns. Anything can grow there. And if you look at the bacteria, the colonies of bacteria, they look sort of yellowish, but there's sort of a greenish tint to them. With plasmid number 2, this has an ampicillin resistance plasmid. So you would have gotten colonies on an amp plate that was plus plasmid. But again, if you didn't use the plasmid, the bacteria shouldn't have been able to survive on ampicillin, so it's clean. Also clean on the kanamycin plates. Again, won't survive on kanamycin because it doesn't have the plasmid. And again, you'd have lawns. If you use plasmid 3, that is a plasmid that has a resistance gene for kanamycin. So you wouldn't have had any survival on the ampicillin plates because, again, they don't have the gene for ampicillin resistance. If they were plus plasmid, you would have hopefully seen some colonies on a kanamycin plate. And of course, if it's minus plasmid, no resistance gene, it's clean. And ideally, again, on your LB plates that are just food, you'd see lawns. And in either case, for plasmid 2 or 3, They're sort of yellowish, whitish colonies in any bacteria that you grow. So that's ideally what you'd come up with. Now, in terms of looking at what's present, plasmid 1 would have the ampicillin resistance gene and the green fluorescent protein gene. Plasmid 2 would have the ampicillin resistance gene, but not the kanamycin resistance gene or the green fluorescent protein. And then plasmid 3 would have the kanamycin resistance gene. So again, this is what would have turned up ideally. Now, will that happen for you guys? I don't know. Some years we're batting 1,000. Other years, some groups have some trouble. And this year I feel like all bets are off with everything, right? So now, what happens if you have plasmid 1 or you have the AMP resistance or the AMP plate with some glowing bacteria? How can you observe that? Well, the bacteria glow under UV light. So we have a UV lamp here. There's a red button. You'll push this to turn it on. You can feel the lamp vibrate when you switch that on. Now, there's a metal sleeve here that slides. There are long waves and short waves of UV light. The short waves are okay to be exposed to. I'm sorry, take that back. The long waves are okay to be exposed to. The short waves can be damaging to DNA and vision. So I've got this taped into position, so you should only be using the long waves. Now, that said, don't turn the lamp up. Don't look at the lamp. We'll keep it facing the table and over the bacteria, okay? So to try and give you a sense of what this will be like, you'll take your plate. If you want, you can flip it so that it's lit up. You'll turn on the lamp, and you can adjust the brightness by pushing the red button multiple times. And you can, take the lamp off and shine the UV light over the bacteria, and they should glow, okay? Now, to see this, we're going to have the lights off in the room. So what I'd like people to do is make their observations about the bacteria, and then when groups are ready, Mr. Prinz, the substitute teacher, will flip off the lights, and the room's going to get pretty dark. But you can come up and use the UV lamp. Again, keep it face down so you're not looking at the light. Hold it over your Petri dishes. And again, you can remove the lid, just hold it and don't set it down on the table. And you can shine that light on it. This would be a perfect opportunity if you do want to take some pictures of some glowing bacteria. Now's a time to do this. Period 7, group number 5. Looks like you've got some glowing bacteria here, so this will be good for you. So again, everyone will turn off the lights and have their plates up here. You can take this, hold it up, shine the UV light, and you should see some glowing bacteria. If you happen to not get any growth, I'm sorry for that. I will say that groups can share so that everybody will see the glowing protein. So hopefully in every class there's someone that's got the glowing protein that you can look at. I also put together a demo. I put together a demo Petri dish that has the green fluorescent protein, so I'm hoping that works. I put the demo dish. It's just a single Petri dish at the top shelf. So if your group happens to have one that doesn't work, you can check the demo Petri dish, and hopefully that one has the green fluorescent protein and glows. Okay, so that's making your observations. What do you do when you're done? Well, you're going to have these Petri dishes with these bacteria. And what you can do is I'll have trash bags at the lab stations around the room. You can place the Petri dishes in those trash bags, and then I'll have some disinfectant spray out. You can just spray the Petri dishes as like an extra layer of security. And then when I come back in on Thursday, I'll dispose of everything. So have your stack of Petri dishes. You can tape them up if you want, set them down in the trash can, spray with disinfectant, and you're good to go. You can also wipe down your lab station, and of course, wash your hands. Wash your hands before and after. Now, we will have a lot of bacteria growing in these plates on some of them. So I do have some gloves up on the demonstration desk. If you want to use some disposable gloves, you're more than welcome to do so. All right. Okay. Stuff to answer in the packet. So if you wouldn't mind looking at the lab packet, I think we were on page S9. So S9, number 1, just basically complete the chart on page S15. You get your point for that. Number 2, you can answer that, make that like a little 3D point. Okay, if we move on to page S10. S10 is a good, like, for instance, what would you have expected? Okay, looking at these plates, looking through them, is there something that seems like it shouldn't have happened? Okay, or if there's something that seems awry, you'll note that and then explain what seemed to go wrong. So that's a good practice. So do questions 3A, B, and C on page S10. So definitely complete that. All right, page S11. There are a couple of things that you don't need anymore in terms of preparation for the AP Biotest. So questions 4 and 6, scratch those out. So please don't mess with 4 and 6. You will do question 5 though. And let me give you the equation for question 5. Sorry, I forgot to write this down earlier, so helpful timing. So let's say page S11, question number 5. The equation that you'll use is concentration 1 times volume 1 equals concentration 2 times volume 2. So they'll give you 3 out of the 4. You plug and put things in. Make sure you have your units. Okay, so that's question 5. Let's see. On page S12, you'll do 1 through 5. You may look a couple of things up on the internet. So you'll complete that. And then just make sure you got that table completed on S15. So that should walk you through the packet. The packet we do on Tuesday when we get back. If you have any questions, let me know. Just make sure you keep things neat and tidy. You know, keep your hands clean. You know, keep lab stations clean. Record the information up here. Shoot me an email if you have questions. But I think that should take care of everything. I hope things go well for you. I hope stuff glows. And I hope you have a great break. So everybody take care. I'll see you soon. Bye-bye. Got to find the off button.